Good evening and welcome. I'm Mike Botchin. I'm also a professor in molecular and cell biology. And today with Matt, Samantha, Andrea, and Noah, I'm delighted to kick off our talks. We've had a series of these virtual events during our shelter in place, and I've been really pleased with how well they've gone. While nothing takes the place of personal and live interactions, these Zoom meetings have allowed us to stay connected with each other and, most significantly, our alumni and friends around the world. We've called tonight's event Rising Stars. Interesting title. In many ways, Berkeley's history can be told as the story of paradigm-shifting discoveries. Immuno-oncology started here. CRISPR genome editing are examples of the type of discoveries that have come out of our division. How have we done this? Well, we recruit amazing people, and we are exceptionally good at basic science. Basic science is the theme that's run through our virtual events this fall, these series this fall. By basic science, we mean fundamental or foundational, not simple, but the basis. I don't think there's a Nobel Prize in physical or biological sciences today where basic science hasn't been primary. And now that we're on the other side of the 2020 Nobel Prize season, it only strengthens that statement. But really, I want to go back to how we do this, how we manage to stay at the forefront, which is that, simply put, we recruit amazing people. The researchers you'll hear from tonight represent the potential that Jim Allison won the Nobel Prize for, Jennifer Doudna did when they were rising stars. Recruiting faculty is one of the best parts of the job that I have as dean. And when I thought about who feels equally committed to new faculty, graduate students, and faculty mentorship of students and each other, I thought of Matt, an accomplished researcher in his own right. Our moderator this evening is Matt Welch, a professor in the Department of Molecular and Cell Biology. He leads a lab that's trying to understand how microbial pathogens target the machinery of the host cells during infection, with the overall goals of revealing crucial mechanisms of infectious disease and uncovering fundamental mechanisms of the inner workings of our cells. Currently, he's working on the regulation of actin assembly, cell migration, interaction between pathogens and the host actin cytoskeleton, developing drugs that target the cytoskeleton of parasites. Okay, Matt, take it away.
All right, thank you very much, Mike. It's a pleasure for me to play a role in today's event. I'm also really excited to hear about the work being done by my excellent colleagues. Just as an introduction to me, I've really been hooked on science ever since my father, who was a mathematician, worked at IBM Research in New York, took me into his lab on the weekends when I was a kid. And I'm really fortunate that throughout my education and training, I've only attended outstanding public schools and universities, including the University of Michigan as an undergraduate, and here at UC Berkeley as a graduate student, and then over across the Bay at UCSF as a postdoc. And I've always really been grateful for the amazing education and training that I received, and I'm also committed to public education and research. And of course, I feel very privileged to be working here with the amazing colleagues I have at Berkeley. So as you know, science takes dedication and collaboration and cooperation and creativity. And in listening to our speakers, I'm pretty sure that this is going to come through in spades. So now I'm going to introduce you to three of our rising star researchers who are doing really important work here at Berkeley. We'll delve into their research discoveries, and we'll also talk about some implications of their work in the future. So I'm always really impressed by the diversity of basic research topics that are being addressed by our faculty. And of course, this young group is no exception to that. This evening, we're going to hear about research on all biological scales, ranging from the very small scale, subcellular, and even molecular scale, to the larger organismal scale. And we're going to hear from Samantha Lewis about the inner workings of subcellular structures involved in energy production. And then we're going to hear from Andrea Gomez about the interactions between cells in the nervous system. These are called neurons, and we'll learn about how these enable computation in our brains. And then finally, we're going to hear from Noah Whiteman about how interactions between species, in this case, insects and plants, influences the evolution of both types of organisms. So I'm also, of course, very impressed with the overall diversity of this group, and I hope you will be as well. It's so important for our students that the diversity of our faculty really reflects the diversity of our student body and also of the people of California. So I'm now going to ask Samantha, Andrea, and Noah to tell us about their research in general and also their current projects more specifically. We're going to leave time after each talk for questions from the audience, so please feel free to post your questions in the chat. We may not get to all of your questions, but we'd really like to hear from you, especially from those of you who are not scientists. So now let me take a moment to introduce Samantha Lewis. Samantha is an assistant professor of cell and developmental biology in the Department of Molecular and Cell Biology. Her research focuses on the biogenesis of mitochondria. These are organelles that play a really important role in cellular energetics and metabolism. Mitochondria, some of you may know, are derived from bacteria, which in an ancient endosymbiotic event that occurred more than 1.5 billion years ago, became organelles inside of the eukaryotic cells. These are our cells. And thus mitochondria have their own genomes. And so Samantha's interested in mitochondrial genome inheritance and replication, as well as mitochondria assembly, cell metabolism, the interactions between mitochondria and other membrane-bound organelles. So Samantha, I'll turn it over to you. Can you tell us a little bit more about your research and the projects that you're currently working on?
Thank you, Matt. Thanks for that wonderful introduction. I'll just share my screen. All right, can you see it? All right, here we go. Again, thanks, Matt, for that great introduction, and thanks to the organizers for hosting us here this afternoon. I joined the Department of Molecular and Cellular Biology this year, and I'm really excited to tell you a little bit about my past work, my research group, and what we're hoping to accomplish here at Berkeley. I'll start again by thanking the audience for joining us remotely during what's kind of a tumultuous time. While we might be physically distanced, we're all linked by this love of biology and excitement about future discovery. And in fact, what I'll tell you tonight is one way that that need for connection, that reaching out for connection, is a part of the very structure of our cells and how that's necessary for cellular function. So as Matt mentioned, I study mitochondria, and on my slide, you can see a cartoon of a eukaryotic cell where mitochondria are labeled in blue. In the nucleus, which contains most but not all of the DNA in our cells, is this big green circle kind of towards the center. You might know of mitochondria as the powerhouse of the cell. So these are parts of our cells that break down fats and sugars that we ingest to produce chemical energy that our tissues can use. And in that process, produce CO2, which we breathe out via respiration. So mitochondria contain their own genomes, as Matt mentioned, which are represented here on the slide by these little green dots that are along the mitochondria. And there are sets of genes in both the mitochondrial DNA and in the nuclear DNA that are required for mitochondrial function. So in my group, we use fluorescence microscopy to visualize mitochondrial behavior in living cells and tissues to better understand how they maintain their mitochondrial genomes that are so important for function. So here I'm going to kind of transition from this cartoon representation to a real image of what mitochondria and their genomes look like inside of our cells. So here again, the mitochondria are in blue and DNA is in green. And there's this big nucleus that contains most of our genome over onto the left side of the screen. And then there are many green dots that are along each of these blue filaments that are the mitochondria, which represent mitochondrial genomes. And what I hope you can appreciate is that there are many, many mitochondrial genomes in the cell, and that they tend to be evenly distributed along the mitochondria. And this is really important for their function because it's the local production of the gene products that are in the mitochondrial DNA that contribute to respiration. And so one question that I have been interested in my career is what happens when mitochondrial DNA localization or synthesis are perturbed. And we know from patient studies that there are diseases that arise from this happening. So mitochondrial diseases are very highly variable. They're progressive diseases that are degenerative, and they affect different tissues in different ways. There are currently no treatments for mitochondrial diseases. There are no cures for mitochondrial diseases and limited treatments. And so one of the things that has driven me in my research is to better understand the underlying mechanisms by which mitochondrial genomes are maintained, because I think by doing that we could understand pathways that could be targeted for the development of therapeutics that could treat mitochondrial disease. And this has much broader implications than just for those who might have a hereditary mitochondrial disease, because mitochondrial function is really critical for healthy aging. So we know that mitochondrial function tends to decline in normal individuals in muscle and in neurons, and that this can cause aging-associated common diseases. And so mitochondrial function in aging has been linked to Parkinson's disease, for example. It has also been linked to sarcopenia, which is aging-related muscle loss. And so one of the things that I would like to do with my group here is to better understand why mitochondrial DNA maintenance and why mitochondrial function tend to vary between different tissues and how that might contribute to the onset of these aging-related diseases. So in my past work, I used fluorescence microscopy to visualize mitochondria inside human cells that are living in a petri dish. So here you can see a movie of one cell where the mitochondria are labeled in blue. And what I think you could appreciate here is that the mitochondria are moving, they're dividing, they're fusing, and they're constantly active. And so I wanted to try to address the question, how is it that there are mitochondrial genomes that are evenly distributed throughout all of the mitochondria of the cell, given this high level of activity? And in this past work, I used different reporters for mitochondrial DNA synthesis and mitochondrial division to capture these processes in living cells. And what I discovered was that the locations where mitochondria tend to divide were also the same locations where the mitochondrial genomes were replicated. And it turns out that this is very important for maintaining that distribution of genomes throughout the cell. And so here again, I'm showing an image of mitochondria in blue in a cell, and this is kind of like a slice through a cell where the nucleus is towards the bottom of the frame and the edge of the cell is towards the top. In a second discovery, what I found is that there are actually interactions between different organelles in the cell that regulate this process of mitochondrial division and associated mitochondrial genome replication. So here, I'm going to add onto this image another organelle, which is labeled in red, the endoplasmic reticulum. And what you can see is that the structure of the endoplasmic reticulum, or the ER, is very complex. And there are also many positions where the ER membrane and the mitochondrial membrane come into close approximation with each other. And indeed, some of my colleagues made a really critical discovery that the endoplasmic reticulum actually directly interacts with mitochondria and tends to wrap around mitochondria, kind of like a handshake, to constrict mitochondrial tubules. And what I found was, building on their work, was that it's exactly at these sites where the organelles are connecting with each other, they're interacting with each other, where mitochondrial genomes are synthesized. And so by looking at these locations where mitochondrial genomes were being replicated, I could predict that there would be a contact with this other organelle at the same site, and that ultimately this would lead to mitochondrial division. So the take home here is that mitochondria contain their own genomes. The maintenance and distribution of those genomes is really critical for cellular function and human health. And it's these interactions or connections between organelles, these organelles that may be in communication with each other, that are really important for maintaining the mitochondrial genome. And so moving forward in my group here at Berkeley, one of the questions that I'd really like to address is, what are the cellular mechanisms that underlie the tissue-specific differences in mitochondrial diseases that I mentioned? And there are a few different ways that we're doing that. Of course, we're building on our tools using live microscopy to examine mitochondrial DNA distribution and synthesis in cells. We're also taking advantage of the environment to use CRISPR gene perturbations to map out the genetic pathways that are required for mitochondrial DNA synthesis, and also for maintaining these interactions between the mitochondria and the ER. And then we're also using single cell gene expression studies to understand which cells in an intact animal are using which pathways and which pathways are important in which tissue types for maintaining mitochondrial DNA. And to do this, we're using a model nematode, which is a very small worm, Centrobditis elegans. And so I have some talented students in my lab who are adapting the microscopy approaches that we used in cultured cells in a petri dish to actually examine mitochondria and their genomes and these processes of organelle interactions in living C. elegans. And by doing this, what we can learn is how the different tissue types or different cell types in the same animal are reacting differently to perturbation of mitochondrial genome synthesis. And we can also use this model to test potential therapeutics to understand better which types of treatments might specifically promote mitochondrial genome maintenance or quality control in, for example, muscle or in neurons, as opposed to other tissues where it seems like those things are not as important. So with that, I will thank you for your attention, and I'm ready to take your questions. Thanks.
Thank you so much, Samantha. That was wonderful. I hope that people will, if you have questions, please go ahead and type them into the chat. While I'm waiting for your questions, I do have a couple of questions for you, Samantha. First of all, you said that different kinds of cells and different tissues have different mitochondria. So could you elaborate on that a bit? Why do you think that different kinds of cells and tissues have different kinds of mitochondria?
That's a great question. So what we have and others have observed is that different cell types and different tissue types, their mitochondria use different substrates for energy production. So some rely more on breaking down fats, some rely more on breaking down sugars, and that this is linked to differences in the shape of the mitochondria. So if in the worms, what we can see is that in the same individual, the shape of the mitochondria in a muscle cell is very different than the shape of the mitochondria in an adjacent hypodermal cell, for example, or in a neuron. And so what we would predict, because of this link between the dynamics and shape of mitochondria and the genome maintenance, is that this will also play out in terms of the number and distribution of the mitochondrial genomes between different tissues.
Very interesting. The other question I had is, how you said you may be able to develop therapeutics that target mitochondria. So how could you imagine that this would happen? How could you develop a therapeutic that would treat a disease caused by mitochondrial dysfunction?
Oh, that's another great question. So one of the things that we have observed already in the worm models is that there are certain tissues where mitochondrial DNA copy number tends to decline with age. So the abundance of those genomes that are so important tends to go down with age. And whereas there are other tissues where that does not happen. And so one of the things that we'd like to explore is to try to identify like potential therapeutics or small molecules that could boost the mitochondrial DNA copy number specifically in the tissues where that is needed. And so that type of experiment, it could play out in terms of using a library of drugs that could be repurposed for stimulating mitochondrial genome synthesis by targeting one of these genetic pathways that we're able to identify using the CRISPR-based perturbations.
And we do have a couple of questions from the chat now. The first question is from Tom Klein. Can you remind us why mitochondria even bothered to retain this vestigial genome from when they were bacteria originally? And when most of the genes have been sort of migrated to the nuclear genome?
That's a great question. Yeah, so mitochondrial genomes, the thought is that in the past, mitochondrial genomes were much larger than they are today and contained more genes, including potentially genes that were necessary for their own synthesis. And now most of those genes have been transferred to the nuclear genome. So there's only a small subset that are retained in the mitochondrial genome in our cells. And it's really a key question. What is different about those genes and why haven't they been transferred? And there are several different thoughts about this in the field. One of them is that there's actually something about the local transcription and translation of those genes that is necessary for their function. So mitochondria have their own ribosomes, and it could be that those transcripts need to be processed and then translated specifically inside mitochondria to have their function. Another thought is that those genes could be key to regulation of overall mitochondrial function and respiration, and having those situated locally provides an extra layer of control and ability of the cell to respond to local changes in respiration rates.
Oh, thanks. We do have a question from the general audience too, which is, why are diseases that attack mitochondria hard or harder to cure maybe than other diseases?
So mitochondria are so important for our cellular function that it's actually really hard to do some genetic studies because a lot of the perturbations are just lethal. And so, we perturb a pathway and the cells just die and we can't even do any studies with them. Another reason is that all of the current gene editing technologies are applicable to nuclear genomes, but not necessarily to mitochondrial genomes in animal cells. And so what that means is that we're kind of limited in our ability to manipulate mitochondrial DNA genetically to try to understand what are the key features that could be targeted for treatment. And this is another area of research that I'm very interested in my lab, is to try to develop or adapt gene editing platforms that would be applicable to mitochondrial DNA in animals.
Okay, and I think we have time for one more question here, which is a question about the effect of physical exercise on mitochondria. So it's the question is, what about nuclear genes that regulate the dynamics of the mitochondrial reticulum? Physical exercise maintains mitochondrial mass without activity. Any ideas why?
So what I know about human physiology is that exercise does stimulate mitochondrial behavior, fusion and fission dynamics, and stimulate the production of more mitochondrial mass. And so one of the ways to get around that disease state that I mentioned, sarcopenia or muscle loss in old age, is to exercise more, basically. So you have more muscle mass, which means more mitochondrial DNA in our case. I do think that that there are behavioral effects that are going to be impacting these pathways for sure.
It looks like we may even have time for one more question. You mentioned changes to mitochondria with aging. So does the mitochondrial genome then change over time as we age? Or is it something else about the mitochondria that's changing?
Oh, that's a great question. So I think there are lots of different things that can change about mitochondria with age. And just one, for example, would be lipid oxidation status. So the composition and oxidation of lipids that build those mitochondrial membranes can change with age. And that can also have effects on the contents of mitochondria, including mitochondrial DNA. I'm not sure exactly what could be changing about the DNA itself with age, other than we already know that the number of mitochondrial genomes per cell does tend to decline in some models. But it's a really great question, and something that we would love to explore further is to look more at a molecular level at mitochondrial DNA over a time course of aging.
All right, thank you so much, Samantha. That was such a pleasure to listen to your, hear more about your work. I'm going to move on now to our next speaker, who is Andrea Gomez. So just a little bit of introduction to Andrea. Andrea is an assistant professor of neurobiology in the Department of Molecular and Cell Biology and the Helen Wills Neuroscience Institute. And her research focuses on organizing signals that sculpt synapses, which, for those of you who are not familiar, are the junctions between nerve cells. Her group is investigating alternative RNA splicing as an organizing signal. This is an RNA processing mechanism that increases the coding power of our genome. And linking RNA processing and synaptic function is critical because aberrant splicing and synaptic dysfunction underlie many conditions, such as autism, intellectual disability, and neurodegenerative disorders. So Andrea's team is using a range of different techniques to decode the cellular rules that instruct and maintain synaptic organization. So Andrea, can you tell us a little bit more about your work and the projects that you're currently working on?
Yes, I guess I can. Can you see everything correctly?
Yes, we can. Looks great.
Yeah, so my goal today is to give you a peek into my brain and tell you a bit about what I think about all the time and what me and my members of my lab here at UC Berkeley dedicate our time to, which is to understand the organizing signals of the brain. We ask, what is the nature of these signals and how do they organize the brain? How do they organize the connections between neurons, which many of you know as synapses? Really, what information in our genome, in our DNA, instruct for the organization and neuronal activity that is responsible for our thoughts, our memories, our feelings, and our behavior. Now, how do we even begin to ask how an overwhelmingly complex structure such as the brain is organized? Well, I look to RNA for the organizing signals, and if there is one thing I want you to remember today, is that the complexity of our brain is matched by the complexity of our RNA. Now, my lab focuses on how RNA diversity is generated in the brain, and we link RNA diversity to synaptic function. To achieve this feat, we combine sophisticated genetic strategies, the latest deep sequencing technologies. We measure the electrical activity of neurons and the activity exchanged between neurons. And we also image the sites of the electrical activity, all of which is required to understand the architecture of the brain function. So what is this image that you have been looking at? Well, this is an entire mouse brain where we have genetically labeled a specific region called the hippocampus. Here that pops up this bright pink. And the hippocampus is a region that is crucial for memory and spatial navigation. Now here is another view of the hippocampus, except now you can appreciate its beautiful connectivity. Now, when we think about activity propagating through this circuit, and it does so in this general direction, in a loop, we know that it is not continuous. In fact, it is interrupted at synapses, at the connections between neurons. It is interrupted as activity passes from neuron to neuron to neuron throughout the circuit. And this is why I love synapses, because they profoundly affect the way activity travels through neuronal circuits. Now, if we zoom in on a single neuron, we can appreciate that it is targeted by many different types of neurons with different types of synapses, and it gets targeted with high precision. But how do these neurons know how to connect to each other? How do they know where to connect to each other? And how do they change and modify throughout our lives? Well, I'm a synaptic biologist and my training is in developmental genetics. And the way we think about these types of questions is the following. As activity passes from neuron A to neuron B, once it reaches the end of the axon, the activity triggers the release of chemical signals or neurotransmitters. These neurotransmitters travel across the synaptic cleft and bind to receptors on the next neuron, which open up channels and a wave of positively charged ions rush in, thus continuing the propagation of activity. And this process repeats over and over again in the networks of the brain. Now, my work focuses on a class of proteins called transsynaptic organizers, which serve as molecular signaling bridges, one made by the presynaptic cell, the other by the postsynaptic cell, such that when they're bound, they bring together all the components for synaptic transmission. They bring together the synaptic vesicles packed with neurotransmitter, and they also cluster the receptors on the other cell so that they are directly opposed to the synaptic vesicles. Now, there are many genes that encode for these transsynaptic organizers, and we can imagine that they can be mixed and matched in combinatorial ways to generate all the different types of synapses that we observe in the brain. However, when we use this logic, we run into somewhat of a coding problem, which is how many different combinations of transsynaptic signalers do we need to generate all the synaptic properties in the nervous system? And to illustrate my point, we have 20,000 genes, mice have 20,000 genes, even the worm C. elegans has about 20,000 genes. However, humans with our 90 billion neurons make anywhere from 600 trillion to 2 quadrillion synapses. Mice also have a lot of neurons, 75 million neurons, making about 500 billion to 2 trillion synapses. And the worm with 302 neurons and 7,000 synapses, which tells us that the number of genes does not scale with brain complexity, which is why I'm interested in RNA alternative splicing. So what is alternative splicing? Well, if you recall the central dogma, you remember that DNA gets read into RNA and then RNA gets read into protein. However, when RNA gets read from DNA to RNA, it is not ready to be read into protein. And the reason is because DNA contains exons and introns, which get directly copied into RNA. Now, the exons are what we call the coding part, which contains the info to become a protein. However, the introns, or the non-coding part, needs to be cut out. So the cellular machinery called the spliceosome, which as the name suggests, cuts out the introns and splices together the exons. And with this simple cutting pasting mechanism, we can essentially bring together the exons together and cut out the introns. Now, what occasionally happens is that instead of going in order from exon to exon, what can happen is you can get an exon skipped. And essentially, this coding part of the exon will get entirely skipped, and this exon will be adjoined to this exon, creating a completely different version of the gene. And with this simple cutting pasting mechanism, we now have a way to make many versions of the same gene. Or in other words, alternative splicing can expand the coding power of our 20,000 genes. And compared to any other tissue in our body, our brain exhibits the highest degree of alternative splicing, and you may not be so surprised that alternative splicing is highly dysregulated in neurological disorders. And I hope you can now appreciate that the extent of alternative splicing scales with neural network complexity. For when we compare animals, as brains become more complex, we observe higher amounts of alternative splicing. In fact, the highest degree of alternative splicing is observed in primates. So not only can this mechanism expand molecular complexity, but it has the potential to generate a large diversity of synaptic organizing signals. And in 2016, I made an important discovery, which is I identified an alternative splicing program that specifically targeted transsynaptic organizers. And by mutating a splicing factor called SLM2, where we, in the mutant, we lose multiple versions of a gene called orexin. What we saw next was pretty remarkable. One is a single exon change dramatically changed the way that mice think. They exhibited a lot of different behaviors in paradigms where animals can recognize different objects and identify novelty. And when I probed a little bit further, I also saw that synapses in the hippocampus also dramatically change. So here I am recording from a neuron and I'm stimulating the axons and observing the response. And what you can see is that in our SLM2 mutant animals, we see a dramatic increase in synaptic strength. Now, this dramatic increase in synaptic strength was also associated with a complete loss of synaptic plasticity. So with that, I hope that I convinced you how exciting and how important alternative splicing in the brain is, or rather how important it is to brain complexity. Now, with RNA processing in mind, I will continue to explore exciting new areas in my lab, including input-specific processing. How does a neuron know that it's receiving information from one brain area or the other, and how does it respond accordingly? Considering we know that psychedelics induce large-scale plasticity events, how does this change the diversity of RNA in the brain, and how do these changes in RNA change neural circuitry? And also, we're looking to observing how these splice codes are in the central nervous system in our brain, but also how these splice codes are in our gut brain or in our enteric nervous systems. And with that, I want to thank the members of my lab, but especially the Department of Molecular and Cell Biology and the Helen Wills Neuroscience Institute, from which I belong. And with that, I am happy to take any questions.
All right, thank you so much. So interesting. So while we wait for a few questions to come in through the chat, I have a couple of questions for you. I'm just obviously we're all intrigued by the fact that the brain is so amazingly complex. You've talked about a quadrillion, possibly 2 quadrillion synapses, and then we've got all this extensive alternative splicing layered in. So what do you see as some of the key approaches that can really help move us forward and to make sense of this complexity?
Yeah, I mean, what I've learned, like kind of as I span molecular to circuits, is that at every single level, these systems, they're highly modular. And because of this modularity, when we perturb say one aspect at one level, let's say the RNA level, it actually generates sometimes a stereotyped behavior at the next level. Now, while we can't linearly say these are the things changing, the fact that these stereotypes exist actually gives us a lot of access for understanding the relationship between, say, RNA and synaptic function and circuit function.
Mm-hmm.
Now, kind of going to, what I think is critical is, I think the way, at least the way I was taught about RNAs, I really did not appreciate truly just how amazing and diverse RNA can be. And I think deep sequencing and select populations of neurons is going to be absolutely critical, and not just looking at the identity of genes that go up or go down during plasticity events, but really looking at the kind of at the resolute scale, at the fine scale, how RNA is encoding for selective aspects, how these exchanges of or incorporations of exons, how 5' and 3' UTRs are changing upon plasticity events in the brain. So because of that, I think that's kind of a nice strategy. Looking at cell type approach is kind of what I say, a modular strategy. It allows us to kind of ask these type of questions, perturb things in very specific ways, look at how these different cell types are participating, and what are the RNAs that are expressed in those cell types.
Mm-hmm. Mm-hmm. We have a lot of questions coming in, so I'm not sure that we'll be able to get to all of them. One question is, as the brain gets bigger in different species, does it get more different cell types, or just more of a set that's present in animals with less complex brains?
Yeah, and so I guess one kind of caveat of in more complex brain is it also is associated with higher tissue complexity. So there are a lot more cell types observed in, say, primate brains compared to rodent brains, and so on and so forth. So the specialties, the special neurons that have a specialized function, there are just simply more of them in more complex brain structures. Mm-hmm. If you don't mind, I'll just go ahead and answer the questions, or would you like to moderate?
Oh, I guess let me moderate.
Okay.
Well, we only have time probably for one more question here. If there's one, you want to pick one? I was going to pick I didn't know what RNA splicing was more variable in the brain. Why do you think that is?
Yeah. This is another kind of interesting factor about brain tissue compared to any other tissue in the body, which the neurons in the brain are post-mitotic. So they're no longer dividing. And part of the offshoot of not having a dividing cells is now you have time to make really long genes. And in fact, if you compare the length of genes in neurons compared to any other tissue in the body, genes in neurons are much longer on average. And so what that correlates to is just simply more exons. So if you have more exons, you have a higher chance or higher probability of having an alternative splicing event. So that's our current understanding of why that is.
All right, thank you. Thank you so much. That was so interesting. And I think we need to move on to the next speaker now, and our final speaker, who is Noah Whiteman. Noah is an associate professor in the Department of Integrative Biology, and his work combines natural history with molecular biology to study the mechanisms underlying coevolution between hosts and parasites, with a focus on plant-animal and plant-animal microbial interactions. And his lab examines these questions using a suite of numerous and multidisciplinary approaches, which I'm sure you'll hear about as we go on. So Noah, please tell us a little bit more about your work and the projects that you're currently working on.
Well, great. Thank you, Matt. Can everybody hear me? Does it look good? Can you see the screen?
Looks great. We can hear you. Thank you.
Terrific. So it's my pleasure to be here. Thank you for that kind introduction. Let's see if I can get this going here. There we go. You can see the butterfly wings flapping. And my laboratory is largely focused on understanding how toxins made by plants, animals, and microbes have shaped the evolution of life on Earth. And today I want to share a relatively new discovery we made with you focused on butterflies. And some of these butterflies evolve bright colors to attract mates, but others, like this monarch butterfly, evolve bright colors to warn predators of danger that lurks within. And by this month, as most of you know, the monarch butterflies that were born in the eastern part of North America, east of the Rocky Mountains, from Georgia to Ontario, all the way west to Colorado, have reached the OML fir forests in the mountains of Michoacan, Mexico, nearly 10,000 feet above sea level, where they will spend the winter. And they were born in the prairies of North America, and they will return to the prairies of North America next year, some of them making a 6,000-mile round trip, which is really incredible. Numbering in the hundreds of millions, these butterflies flew all this way to this one spot where they've never been before. And this is truly a wonder of the natural world. And one of the things I'm going to share with you today is some insight into what enables this migration. How do these butterflies make such a brazen journey during the day, unscathed by the flocks of hungry birds that we all see around us? And the secret was revealed in the 1960s by Lincoln Brower and colleagues, who showed that naive songbirds like this blue jay would at first eat monarchs readily, but then would vomit, suggesting that monarchs were actually very unpalatable. They would then avoid monarchs in the future, learning to associate the color pattern with this unpalatability, helping explain how the bright orange coloration evolved to warn predators of chemicals lurking within their beautiful wings. And it was Miriam Rothschild and colleagues who discovered that heart poisons called cardenolides, or cardiac glycosides, were actually in the bodies of these butterflies. And most amazingly, they found that the toxins were actually obtained while the butterfly was a caterpillar. Here's a monarch caterpillar munching on a milkweed leaf from their host plants on which they're specialized. And remarkably, these cardiac glycoside toxins evolved many times independently across plants and even in some animals, from milkweeds on the left to foxglove. Here's the jade plant family. Here's a lily, lily of the valley. Here's a wallflower from the mustard family, adonis from the buttercup family, and maybe even a shrub that you walked by today. And quite simply, these plants evolved the ability to make these toxins that was selected for to prevent animals from eating them. So the idea is that these are natural insecticides. And you may have read about these toxins before in Agatha Christie's books. Agatha Christie was a pharmacy worker during World War II and knew about the toxic properties of plants such as foxglove, and often used them as murder weapons in her books. Digitalis was actually the subject of the first clinical trial in 1785 by William Withering, who found it could treat dropsy, also known as congestive heart failure. And even my own grandmother, this is a picture of her on her wedding day, was on digoxin near the end of her life. And I'll show you a little video of how digoxin actually works to cure dropsy. So the drug blocks the sodium potassium ATPase, which is also just called the sodium pump. And you can see it here in the membrane of a cardiac cell. And this pump is very important because it regulates the ionic gradient, in particular, the amount of sodium and potassium inside and outside the cell. And that has major impacts actually on the resting membrane potential, but also in the case of the heart muscle, because when that pump stops working, you'll see in a minute the digoxin, that's the plant toxin right there, landing on the first extracellular loop of that pump, it stops the pump from working. And what happens then is that sodium builds up inside the cell, potassium outside the cell, and that causes the sodium-calcium exchanger also to stop working. So then calcium builds up in the cell, and you get a stronger strength contraction of that heart muscle. A little too much though, and of course you get arrhythmias and the animal will die, but a little bit is a drug. And so it's also the herb of life. Now the toxin binds to that first extracellular loop and fits into that pocket. And the question that I hope you're asking is like a hand in a glove, why does it do that? A big clue came in 1991 with the discovery that our own adrenal glands actually seem to make cardiac glycoside-like hormones in minute quantities that our bodies use as a hormone to regulate the sodium pump. And this finding suggests that plants are merely taking advantage of an ancient Achilles heel, the binding pocket for cardiac glycosides that all animals seem to have evolved. And as Agatha Christie said, old sins cast long shadows. And as an evolutionary biologist, I would say that old evolutionary events shared by all animals cast even longer ones. And plants have been giving herbivores overdoses of our own hormones for millions of years. Very clever, but evolution finds a way, which brings us back to the monarch. How do they resist the very toxins that they obtain from their plants that are so toxic to other creatures? And I'm going to tell you a little bit about how my lab focused on the very same spot in the sodium pump that the toxin binds to. And building on decades of research on this pump, we decided to once and for all try to figure out how they resist it. And this is an evolutionary tree showing how the milkweed butterflies with the monarch at the top, the queen, the crow, the orange-winged tiger, and then a relative that isn't a milkweed. You can see in this particular tree, there are numbers here to the right and also over the nodes and the branches. And those are the three places in the sodium pump that we thought were important for binding, and other people had shown that they probably were, at least two of them. And indeed, we found that the three mutations in the monarch were very different from the other butterflies. And those were candidates for resistance against this heart poison because it's in the very same place where that poison binds in our sodium pumps. And now we wanted to know if these mutations were sufficient for the monarch to resist the toxins. And to do that, we settled on a technique used by geneticists that involved introducing each of these mutations into the model animal, the fruit fly, Drosophila melanogaster, which also has a sodium pump that actually is very similar to ours, and to test if we could obtain fruit flies that were like monarchs. In other words, could we turn fruit flies into monarch flies? And here are the DNA sequences to show you the goal, which was to turn this Drosophila melanogaster sodium pump sequence into each of these that had evolved millions of years ago across this span of evolution. Unfortunately, just as we are embarking on this journey, two Nobel Prize winning scientists you may have heard of, colleague Jennifer Doudna and Emmanuel Charpentier, proposed how a bacterial immune system called Crispr could be used to precisely edit the genomes of many organisms. And we settled on using this toolkit, which had not really been used yet to retrace adaptations in animals. And because this is an essential gene in humans and all animals, this proved to be a very difficult task. And when we tried doing this, we failed for about 3 years. And just as I thought it wasn't going to work, we got it to work. And thanks to two postdocs from Berkeley shown here, Mariana Karagiorgi and Niels Groen, and a large team of Cal graduate and undergraduate students, we got it to work. And suddenly we had our monarch flies. And this is a monarch fly in the wing of a monarch butterfly, you sense of the scale. And so we were thrilled with this, particularly because this was the first time that multiple amino acid changing substitutions had been swapped in an essential gene in any animal using CRISPR, to our knowledge. So we had these flies, and here they're listed here, the genotypes. We then subjected them to a variety of challenges to simulate feeding on a diet filled with these heart poisons. In this one, we used a water-soluble cardiac glycoside called wabane, which actually comes from the poison arrow tree, which is in the milkweed family. And we found that even at concentrations that would kill a room full of very large animals, The mutant flies that were the same genotype as the monarch shown up here at the top were not affected at all. Their survival was not affected at all. Although the flies without these 3 different mutations couldn't tolerate the toxin, at even relatively moderate concentrations. And what is more, the intermediate stages here were sort of the intermediate stages of evolution, representing the queen and crow butterflies. And so what this told us was that there were 3 mutations that were sufficient for heart poison resistance in the monarch and enable this journey that these monarchs take of thousands of miles as they carry poisons in their wings, protected from birds and predation. But there's no such thing as a free lunch. And you're going to hear a loud noise, but this is on purpose, so bear with me. So those are some of the flies that we made. On the left is the control. That's a very vigorous shaker. You wouldn't want this to happen to you. Okay, so that fly is walking right away, but check out the flies that got these mutations in the sodium pump. They're having some trouble recovering from that, and that's called bang sensitivity. And because we were able to isolate the effects of each of these mutations across millions of years of evolutionary time, we were able to understand why the ordering of these mutations mattered. And let me explain what I mean here. So it turns out that the very last mutation to evolve, this histidine at position 122, which is only found in the monarch, that particular mutation by far confers the most resistance to the toxin, yet it evolves last. And it turns out that we think is because it is the most problematic in terms of seizures. It causes the most seizures of any of these mutations. But earlier on, a change at position 119 to a serine that evolved millions of years earlier actually rescues the negative effect of that histidine. And so by teasing apart these evolutionary steps using CRISPR, we were able to understand the order of the mutations and why that mattered. And again, we go back to old sins cast long shadows and evolutionary ones cast even in longer ones. And so the physiological constraint imposed by this critical ion pump means that sometimes there's really only one route to the summit of Mount Improbable, as it were. And indeed, other insects adapted to milkweeds evolved the same resistance substitutions independently, suggesting that evolutionary constraint can be quite strong, limiting the number of paths available to reach a fitness peak. So to summarize, we found that 3 mutations in the sodium pump were sufficient for allowing these amazing butterflies to sequester toxins they obtain in the northern prairies as they migrate to Mexico and back. And basic discoveries like CRISPR are changing the face of medicine and evolutionary biology, and these 2 fields can inform each other in important ways. Evolutionary constraint provides opportunities for opponents during the evolution of conflict. And even fruit flies can teach us something about ourselves. Mutations in the same gene in humans are known to cause seizures just like the ones we observed in the flies. But by using flies to study how these single mutations affect this sodium pump, we were able to open a window into basic biology of a protein that's critical to the operation of every animal cell. So thanks for listening to this snippet of research, and I look forward to questions.
All right, thank you so much, Noah. That was fascinating. And I think we should also acknowledge that Noah's mom is actually here. And so welcome, Noah's mom. Hopefully you're proud of him.
She's somewhere.
So I have a quick question for you. Before we get started with the questions from the chat. So can you think of examples of human evolution of either toxin resistance or examples of which human evolution has been shaped by toxins?
Sure. So, probably the most abundant gene family in animals and plants are the cytochrome P450s, right? And so, those play a huge role in the detoxification of toxins that we imbibe every day, including things like caffeine, for example, which evolved, it turns out, as an insecticide. And so just like fruit flies, we use many of the same kind of conserved enzymes to convert these toxins into less toxic substances. And so, that's one thing I would say. But the other thing that's kind of interesting is that there's a whole suite of toxins that evolved in plants and fungi that actually mimic these deeply conserved hormones, or at least bind to the receptors that are deeply conserved. Whether it's, opiates from the opium poppy that are binding to the opioid receptors, that it turns out our endorphins bind to, right, which are endogenous opioids, to these cardiac glycosides that may well be mimicking our endogenous cardiotonic hormones that are regulating the sodium pump. So that's one thing, that these ancient kind of choke points really do allow plants to defend themselves quite broadly. But humans like to eat these things that are toxic, right? So caffeine, nicotine, even capsaicin. And so, yes, we use many of the same pathways that these animals do, and that makes them really nice models for understanding evolution.
And are there general principles that you can think of toxin resistance that you're learning from your work?
Yeah, so, one of the things is that our shared history means that evolution is quite constrained. So we like to think of ourselves as being very special, but, we're also very susceptible to many of these toxins here. But one of the things that I think this tells us is that when you have target site insensitivity, which is what, the phenomenon that we were working with, where you have this very conserved protein, when you tweak it to make it, more resistant, there's a trade-off, right? And so I think one of the things we're learning is that anytime you create, some advantage, you're breaking something else. And, organisms are very well integrated. And so evolution has to work to compensate for that. And I guess that's kind of maybe one of the universal things that this is teaching us. And we've seen this in frogs from some of Becca Tarvin's work, that those receptors, they can evolve to be resistant, or those sodium channels, but there's a cost and that has to be resolved. And so, I think that's what we're learning, is that epistasis within proteins is a very important kind of governing principle that maybe was a little surprising, I think.
Maybe one more question. So it is a kind of a related question about why the mutants block the toxic effect, and is it strictly a binding interaction that's being blocked? And perhaps there's more other ways in which toxin resistance can be evolved as well.
Yeah, so the most common way is just to pump them out, right? And so in our kidneys, for example, that's probably, and we showed actually in these two gene families called organic anion transporters and multi-drug resistant proteins that, fruit flies actually have a pretty high basal level of resistance to cardiac glycosides just from having those enzymes around in the Malpighian tubules, which are, sort of homologous to our kidneys. And so we're doing the same thing. So there's this whole set of phase 1, 2, and 3 enzymes that are involved in pumping or transforming these into non-toxic molecules. But if they get in there and then they start interacting in ways that organisms don't want, that's where target site insensitivity and some of these other things come into play. So to Tom's question about, how do we know this? What I didn't show you is we do have brain nervous tissue preparations from these mutant flies where we can isolate the, sort of just the sodium potassium ATPase. We can look at the activity of that pump in a phosphatase assay. And there we see, we can kind of use a Wabang gradient and have controls. And it's very clear that, it's basically blocking the binding of that toxin. But I think to really get it, we would probably want something like a crystal structure if we really, really wanted to show, co-bound, the Wabang binding pocket, a cardiac glycoside.
All right. Well, thank you so much. I think we're essentially out of time now, but I've really enjoyed all three talks. Amazing work being done here. And I'm going to close by handing the floor back over to Mike Botchin, Dean of the Division of Biological Sciences. So Dean Botchin is a professor of biochemistry, biophysics, and structural biology at Berkeley. He received his PhD from Berkeley back in 1972. And was recruited back again to Berkeley in 1980 to become an associate professor in what was then the Department of Molecular Biology. And his scholarly work at the UC has included contributions to virology and also to unraveling the mechanisms of DNA replication. He's a member of the US National Academy of Sciences, the American Academy of Arts and Sciences, and is a fellow of the American Academy of Microbiology. And he chaired the biochemistry section at the US National Academy of Sciences and is still a member of the editorial board of PNAS today. And he's also currently on the medical advisory board of the Howard Hughes Medical Institute. So please take it away, Mike.
Thank you, Matt, and thank you for keeping us to time. It was an amazing group of speakers. And thanks, of course, to Samantha, Andrea, and Noah for sharing your work with us. As a dean, I like nothing more than to hear faculty convey their own excitement about their research. And it's really contagious, and I really felt it tonight. And a great reminder, of course, of how excellent Berkeley is and how many rising stars we have amongst us. I also want to give a special thank you to our alumni and friends for gathering tonight. I risk sounding sentimental, but biology and the types of science questions and discoveries that we've heard tonight, for me, create a sense of wonder. Just as an astrophysicist might feel looking at the night sky, that wonder. Uh, in these uncertain times, it's actually sustaining for me, and I hope for you as well. There's a lot of beauty and truth in just seeing the pictures of organisms or inside the inner workings of the brain or a cell. While it's essential to look for solutions to problems that we're all hoping come soon, like the COVID-19 pandemic or climate change, it goes hand in hand with foundational work that leads to those solutions. And as a friend of mine once said, without basic science and basic discovery, there is nothing to translate. Basic science has always been, of course, Berkeley's greatest strength and must continue to be so, or maybe one of Berkeley's greatest strengths. Your support, your philanthropy, and your advocacy for Cal means everything to us and to the faculty, and faculty like Matt, Samantha, Andrea, and Noah, and their incredible students and postdocs. We're extremely gratified for your continued interest in Berkeley, alma mater to many of you. If there's anything you want to learn more about, or if you'd like to support our work, for which there's a deep need, please be in touch with us. We absolutely want you to be part of an advancing basic science and discovery at Berkeley. So with that, I'll say fiat lux, go Bears, and good night.